Is it okay? It's going. Okay, today the topic is if my computer doesn't explode. Test. Okay, I guess it's working. Today's topic is Lyme disease. Where to begin? So, in around like 1970s, 1976, there is a state called Connecticut. Right? Let's look at that. And in the state of Connecticut, which looks like this, there is a town called Lyme, and there's also a town called Old Lyme. And Lyme's disease was associated with this place. Okay, so I'm going to tell the story of how we figured out. Hi, Yvonne. How we figured out about Lyme's disease. And I've been there. So I used to live in New Haven when I was at Yale, which is about like an hour, 45 minutes from Lyme. So I've driven there just to like look at it, see what it was like. So the story goes is that around this time in the 1970s, there were two mothers from Old Lyme. And these two moms, all of a sudden, they noticed that everybody in their family had joint pain. Okay, so they go to the doctor, and the doctor diagnosed them with arthritis. Okay, and what did the moms, of course, think about that? It wasn't the moms that were necessarily diagnosed with arthritis, it was their children. Why would that be weird? Kids don't normally get arthritis, right? Like kids don't normally get arthritis. Arthritis is like a joint pain disease for like old people. So of course these moms who were right thought this is strange. I don't buy this arthritis diagnosis. And they live next to Yale. So they contacted the hospital at Yale. And they were essentially like, we want to kind of get a second opinion. We want to know what's going on. So they went to the doctor, and the doctor that they saw was this guy named Dr. Steer. And he was a specialist in rheumatology, so like arthritis. He was a specialist in arthritis. And they went in to see this person. And this person took an interest in researching this topic. So I think I have a picture of him. This guy. This is Dr. Steer. Okay. He took an interest in researching this topic because he was interested in arthritis, and he noticed a couple things. Let me just make sure I haven't missed anything before I talk about the things that he noticed. Here's just in terms of geography. So here's New Haven, where Yale's at. Here is Old Lyme and Lyme, and this river, I'm not sure which is the Connecticut River, but there's an important Connecticut River here, which I think is this right here. Okay, so the moms came from around this area. Okay, and there were a couple things that they noticed when they brought their children to this doctor. Okay, the first thing that they noticed was people saw this. Okay, this is what we call the bullseye rash. And this had a name at the time. It was previously described in Europe. And in Europe, they had given this strange rash this name, Erythema migrans. Okay. And in Europe, they knew that this pattern that they saw was associated with ticks. But in Europe, nobody had ever associated this with arthritis. Okay. In Europe, for whatever reason, nobody had ever noticed outbreaks of arthritis. Okay. But they knew about this sort of like this symptom. And Dr. Steer was sort of like, he found out about the descriptions of these symptoms in Europe, and we knew that they were associated with ticks. Okay, a couple other things that Dr. Steer noticed. So I'm just going to kind of like make a list. This was an outbreak of arthritis. Okay. And what I mean by that is it wasn't just these moms with their families who got arthritis. It was like he noticed that it was a bunch of people in this region. It was a bunch of people in this region who got arthritis. So why would that be weird? An outbreak of arthritis. Why is that weird? Well, it's not contagious, right? Like arthritis, you'd think is like damage over a time accumulated like from working your joints or whatever. It's not contagious. It's not a contagious disease. So that's something that was very strange. Arthritis should not be contagious. The other thing that was noticed was what I'm going to call geographic clustering. So that means like in this map, in this area, all the incidences of this disease were in this area. So that's another weird thing that was noticed was that if this was truly arthritis, it doesn't make any sense that there would be this like cluster of arthritis in one strange town. Like that maybe there would be a statistical fluke, but that doesn't quite really make sense. So that was thought to be odd. The third thing that was noticed was that in this outbreak of arthritis, there was what I will call temporal clustering. That means all these cases of arthritis were reported in June and September. So in spring and fall. Okay. So if it was arthritis, you would not expect to see this sort of temporal pattern. Okay. Other things that were noticed. These regions were rural areas. So it was not in the cities that people were getting this outbreak of arthritis. And then the fifth thing was the rash, the bullseye rash. So all this was noticed before we knew what Lyme's disease was. This is essentially like this strange thing is happening. We're getting an outbreak of arthritis. It seems like it's contagious. It's geographically clustered. It's temporally clustered. It's only happening in rural regions, and they get this strange rash. And Dr. Steer noticed that this rash, this erythema migrans, was previously described in Europe, and it was associated with ticks. Okay, so Dr. Steer put 2 and 2 together and hypothesized that his hypothesis was maybe this outbreak of contagious arthritis, maybe it's associated with some tick-borne disease. Okay, so he's the person who sort of like put 2 and 2 together. Couple other things that were known, and this is what was known about this disease that they called erythema migrans in Europe, was that sometimes it was associated with tick bites, sometimes it was accompanied by nerve pain, paralysis, meningitis, but not arthritis. So there was something weird in that whatever this was in Europe, it wasn't generating arthritis. But in Europe, when they got this disease, what the doctors would do is they would prescribe penicillin, and penicillin was moderately effective at sort of like making this go away. So what would that cause you to think? If the doctors were prescribing penicillin when they saw this thing and it sort of made it get better, what would that make you think? Probably bacterial, right? Because penicillin inhibits cell wall synthesis of gram-negative and positive bacteria. The size of the cell wall is different in gram-negative versus gram-positive, but many bacteria, whether they're negative or positive, are susceptible to penicillin. If I'm correct, it inhibits enzymes that do the cross-linking of the cell wall. and it kills bacteria. So yes, exactly what Dee said is that because of the fact that you'd prescribe penicillin and this would get better, it'd make you think it was like a bacterial disease and possibly spread by ticks. So in the original publication, I think they called this Lyme arthritis. And they suggested it was spread by ticks. This is in 1976. Okay. A couple other interesting things about this is there's a river called the Connecticut River. And what was the interesting thing? I'm going to make an analogy here to RMSF, Rocky Mountain Spotted Fever. What was the unique thing about Rocky Mountain Spotted Fever? In the Bitterroot River. What? It was a west-east sort of like dichotomy. Like on one side of the river, they got this disease, and on another side of the river, they didn't get the disease. And they saw the same thing with early instances of Lyme disease. On the east side, instances of this disease were 30 times higher. Now, it wasn't complete. Like you'd still get the disease. You can still get the disease across the other side of the river. But on the east side, it was 30 times higher. So again, this is sort of like, if you know the history, this is kind of like another thing that suggests maybe this is some kind of a tick-related thing. Here's the Connecticut River. Here's a picture of it. Looks like it's this one. So the cases would be here. Okay. So essentially what was known, before I go into Willy Bergdorfer, essentially what was known at this time was, okay, there's this strange arthritis outbreak. It's temporally, geographically localized. It's probably due to a bacterial disease from ticks, but nobody had proved it. Like nobody had proven the case. So this guy, Willy Bergdorfer, he was working at an NIH laboratory called the RML. What's the RML? We talked about the RML. Rocky Mountain Labs. Willy Bergdorfer was working at the Rocky Mountain Laboratory, which was a BSL-4 facility founded in the days of Howard Taylor Ricketts with the purpose of researching Rocky Mountain Spotted Fever. And they were continuing in their research efforts, studying dangerous tick-borne diseases. And the RML, at the RML was this researcher named Willy Bergdorfer. And Willy Bergdorfer, essentially his job was people would send him ticks from all over the world. And he had a microscope and he would just take the ticks and dissect them and look in the microscope in their guts, in their things that are inside of ticks. And he would look for bacteria. And he would just kind of be like, well, let's look and see all the things that are in bacteria and let's see if we can find some new diseases. Okay. So that was essentially like his job description while he was working at Rocky Mountain Labs. So he gets a nice sample from New York, ticks collected from New York. Okay. And they were Ixodes ticks. In the publication, I think it says Ixodes damani. I think Ixodes damani is some kind of like subspecies of Ixodes scapularis. They're deer ticks. So he's looking at this population of deer ticks from New York, and he sees some new things, which are spirochetes. They look like this. These things, if I were to draw them out, these bacteria that look like this, something like this. So the strange bacteria that form these long spirals. And he finds these in these ticks that were sent him from New York. And fortunately, Willy Bergdorfer was reading the literature on tick-borne diseases, and he had seen the studies that were published by Dr. Steer. These publications were in either Nature or Science. They're big publications. And Willy Bergdorfer had read those publications. So he knew that there was a strange arthritis that was supposed to be in the New York area, in the broader, I guess, New England area. And it was thought to be a tick-borne disease. And he found this new bacteria in ticks from that region of the world. So his idea was maybe this thing that I'm looking at under the microscope, maybe this thing is the cause of Lyme arthritis. Maybe. Okay, so he set up a test to prove this. How would you prove this? What? Inject the bacteria from a tick into a human, effectively giving somebody Lyme disease. Who's going to volunteer for that? Nobody wants to do that. That would be unethical. We would not be allowed to do that. But it would work if you did that, sure. So there's a more ethical way to do these experiments. So what he would do is he got patients who were infected with Lyme arthritis, and he would draw their blood. So he would collect blood from patients infected with Lyme arthritis. And since we know so much about immunology now, what would be in the blood that if the bacteria was the cause of this disease, what would be in the blood that would be useful for experiments? If you get infected with a disease, antibodies. Okay, so remember proteins like IgG, IgE, these things that look like these Ys. They are proteins that cause our body to fight off diseases, and they recognize what are called antigens. They're designed to recognize antigens. And if the bacteria, if this spirochete was causing the disease, you would expect that people who had Lyme arthritis would have antibodies that would recognize the spirochete, right? Makes sense? So you can do an experiment once you have this blood, once you have these precious antibodies, you can do an experiment which we call immunofluorescence. How does immunofluorescence work? If you're looking for X and you have an antibody that binds X, you can mix it with the antibody, and then you have secondary antibodies which shine some kind of light or fluorophore, or that's usually how it's happening. And you can do microscopy experiments where you can set up combinations of antibodies that light up when something is there. Okay, so the way that this would work, what would be that he would take blood from people who are infected with Lyme arthritis, he would extract the antibodies from that blood, and then he would take his tick samples that had this bacteria, okay, and he would put the blood with the antibodies. So imagine this was a smear of tick whatever, and here's a spirochete. He would put the blood on that sample, and if the blood had antibodies that would recognize, they would bind to the spirochete, and then all he would have to do is add what we call secondary antibodies. So the primary antibodies would be the ones that recognize the spirochete. The secondary antibodies would be antibodies that recognize human IgG or human IgE. You could make these in either like a rabbit or a mouse, and they would have a fluorophore, something that shines, and they would cause those bacteria to then light up. Does that make sense? Is that clear? Ifan? What's confusing about that? No, I want to answer your question. I want to make sure you understand it. Does this make sense? You didn't follow. Okay. Let me try to explain again. Okay. so what we're talking about here is immunofluorescence. Okay. The purpose of immunofluorescence is you want to look for something. You want to look for where it is. Does that make sense? It's going to allow you to see something. Okay. So if you want to look for something with your eye, in cellular organisms, you can use immunofluorescence. The way that immunofluorescence works is you have a primary antibody. This is a 1 primary antibody, which recognizes the thing you're looking for. I'm just going to say X. Okay. So in this case, the primary antibody was in the patient's blood. And what they're looking for is the bacteria. And if the patient's bacteria has antibodies, or patient's blood has antibodies for that bacteria, it will recognize it. Okay. And then you can add what are called secondary antibodies. Secondary antibodies bind this antibody and they shine. Okay. Causing X to shine. Does that make sense? So wherever X is, it's going to light up. You're going to be able to see it. You're going to be able to see it wherever X is. Okay. So that was the experiment. And these are the results. This is immunofluorescence of what we now call Borrelia burgdorferi, the pathogen, bacterial pathogen that causes Lyme disease. And the antibodies that were found were from patients who are infected with Lyme arthritis. And therefore, if the antibodies in patients with Lyme arthritis are lighting up this bacteria, what did you just prove? The bacteria causes a disease. You just proved it. So Willy Burgdorfer proved that Lyme's arthritis is caused by the pathogen Borrelia burgdorferi, and it was named after him. And then he did some electron microscopy. Here's some electron microscopy of the spirochete, sort of like looking at it with a finer resolution. Does that make sense? Okay. So what happens when you get Lyme disease? You get very tired, and I've had it before, so I'll tell you my story in a sec. You get very tired. Your joints start to hurt, headaches, fever. That definitely all happened to me. And if you catch it early, it's not really a problem. You can take antibiotics and you'll probably be okay. If you don't catch it right away, it can get real bad and it can infect your brain. It doesn't usually kill you, but it really can like mess you up for life. So, okay, I'll tell my story. Let me just make sure. Okay, my story. So I'll tell you how I got Lyme disease. And it was crazy. It was a crazy story. So I lived in Milford, which if you look at the endemic regions of Lyme disease, this is an old map. It's now kind of like these epicenters are like connecting. So this map is too old. I really shouldn't be showing it anymore. But there used to be 2 epicenters of Lyme disease, one in Minnesota, Wisconsin, Midwest, and then the other in New England. And of course, Connecticut is like right around here. So Connecticut is literally the epicenter of Lyme disease. Although now it's, again, these are connected and there's Lyme disease all over now in Pennsylvania, etc. So I lived in Milford, which was literally like 20 minutes away from the epicenter at Lyme. Okay. And in this epicenter, it's so endemic. Lyme disease is so endemic that 80% of Ixodes scapularis deer ticks have Borrelia. So it's almost like if you get bit by a tick in this region, it's like you're really likely to get this if you don't catch the tick early. Okay. Now, what else makes this bad is that, of course, in New England, much of the regions near cities and suburbs have high deer populations because nobody is really hunting the deer in those areas. Okay. And even in worst cases, like where I lived, the neighbors would feed the deer. So the deer would come into our yards. And of course, wherever you find deer, you're going to find high populations of deer ticks. The deer are the adult blood source for the adult ticks. So wherever you have lots of deer, you're going to have lots of Lyme disease. Okay. So that's the context. And I went through this period where I would see tick after tick after tick in this season when I lived in this region. Okay. So I'll tell you the story where the first time I saw a tick was we were driving home and here's our car. My daughter comes out of the car and she's like, Dad, there's a bug on me. Get this bug off. Okay. So I look at my daughter and I found a questing. It was an adult Ixodes. Questing means like it was like walking around. So I found a questing Ixodes on one of my daughters, Elaine. Pulled it off. I was like, okay, it's a bad sign. It's ticks. Okay. The next day, literally the next day, okay, I'm changing my son's diaper and I see this tiny speck on my son and I don't think anything of it. It's just like a dot. Okay. The day after that, the next day, I change my son's diaper again and the dot is bigger. So then I'm like, okay, I look at it closely. Now I realize it's a tick. Now I realize, oh crap, it's been there for 24 hours. It's now engorged. Okay. And it was an Ixodes tick. So I went through, I would say, let me just also say it was an Ixodes nymph, which if you look at this, here's the nymphs, here's a dime. They're literally like a speck. They're really hard to see. And the nymphs are the ones that are spreading most of the Lyme disease. So I went through a catch-22 and I was like, okay, well, what should I do? What do you guys think would be good advice for me? Like, how should I deal with this situation? I'm living in the endemic center of Lyme disease. I found an Ixodes nymph on my son, which had been engorged for 24 hours. What would you do as a reasonable father or mother? Yeah, like you want to go to the doctor for sure. And I essentially like already decided. So I called my friend at the CDC, but even, well, I would say like the doctor has to go through a dilemma. Like the doctor, it's not clear that my son has Lyme disease, right? So the doctor has to make a choice like, should I prescribe treatment and pre-treat this situation, assuming that this kid might have Lyme disease, okay? And I called my friend at the CDC and I was like, well, would it be better to wait and see what happens and then treat it? Or would it be better to just be like, nope, I know what I'm talking about. My son had an Ixodes tick. We live in the endemic region. Just give me the damn antibiotics and I'll give them to him and we won't worry about it. And my friend was pretty much like, yeah, you might as well just do that. So I went to the doctor and I was like, yep, we want the antibiotics. Just give me the antibiotics. I treated him with the antibiotics. Okay. And I felt like that was the best decision because one of the things that made me a little bit nervous is that if we waited to just like see what could happen, about like 10 to 20% of Lyme disease cases, you don't see anything right away. You might not see the bullseye rash. You might not see any symptoms. So my rationale was like, we might as well just treat this. There's really no downside to treating the kid with antibiotics. Okay. So that's what happened. Okay. Now this is not the end of the story, right? I first found a tick on my daughter. Second found a tick on my son. It was engorged. We treated him for Lyme disease. The next day, no joke, literally like the next day, this shows you how tick infested the region we were living in was. The next day, I wake up in the middle of the night and there is a tick, an adult tick. It was an adult dermacenter questing on my back. Okay. So now what should I think? Like what would you guys think if you were in this situation? What would be your thought process? Should I worry about Lyme disease? Yes, why? It's dermacenter. Dermacenter is what tick? It's a dog tick. Dog ticks do not spread Lyme disease. So, and the other, so there's a couple of reasons I don't have to be worried in this case, this next day. One, it's dermacenter. Dermacenter does not spread Borrelia burgdorferi. Two, the tick was not engorged. It was questing. I didn't find it in me. I found it crawling on my back. So it had not fed yet. And if a tick hasn't fed yet, remember what we learned about ticks is that the longer a tick feeds on you, the more likely you are to get a disease. So if you find a tick even before it's even bitten you, you're not going to get a disease. Okay. So there's two reasons I didn't have to be worried. Okay. So I wasn't worried. Okay. But the story is not over. Okay. The next day, literally the next day, I'm riding to work on my motorcycle. Okay. I rode on the freeway. I forget what freeway, but I'd had like a 20-minute commute. And I was riding on my motorcycle, got to work, and as soon as I got to work, I started to feel like lightheaded. So I started to feel like I was going to faint. And I never faint, okay? That would be something that would be very strange. So I was like, okay, this is strange. I better sit down for a little bit. And it went away. But I was like, okay, good thing that didn't happen when I was riding my motorcycle on the freeway. Good thing I didn't faint. And then I come home. I work throughout the day. I come home, and as soon as I get home, I felt a burning on my arm, and I looked down in my arm, and this is what I saw. This is an actual picture of my arm. So this is what I saw on the first day, the bullseye rash, right? Okay. So what's my conclusion? How did I get Lyme disease? What would you think? There must have been an Ixodes that like I never saw. So it was not the dermacenter tick. There must have been an Ixodes that I never saw on my arm as a nymph, which was so weird because, like I said, this was in a period, literally a week, a condensed week, where I was seeing tick after tick after tick inside my house. And I was literally like checking my body for ticks. And even while I was checking my body for ticks, I never saw the nymph that gave me the Lyme disease. So it's kind of crazy. Like even if you're checking, you might not see the lymph. So my conclusion was it was not the dermacenter that I found on my back. It must have been a nymph, an Ixodes nymph that was feeding on my arm that I never saw. That was my conclusion. Okay. So now I went to the doctor. And I was like, this is funny too, because I had literally just been hired here for my job at Auburn. I went to the doctor and I was like, I have Lyme disease. Please give me antibiotics. And the doctor was like, well, what makes you think you have Lyme disease? And then I held up my arm and I was like, here's the Lyme disease. Here's the bullseye rash. I have Lyme disease. I live in the endemic region. Oh, by the way, I'm a medical entomologist. I was just hired as a professor to be a medical entomologist. So like, I know what I'm talking about. Please give me my antibiotics. They gave me the antibiotics. So they prescribed me with doxycycline, and a couple things about it. So I like again, because I was interested in this. A couple things I remember is like I was more tired than you would like ever imagine. Like I was super tired. I would go through like hot and cold flashes, and then as soon as they gave me the antibiotics, like 2 days later, I felt a lot better. But it took forever for this like rash to go away. So this is 4 days after, even after like they put me on a 2-week cycle. So that's 14 days of antibiotics. Even when the antibiotics were over, like the rash was still there. So what I was thinking was, as I was going through this process, I was like, man, that I could see it. That rash is still there. And I'm like, man, my antibiotics are getting low. Like, is this supposed to happen? Like, should I ask for more antibiotics because it's not gone? But then like what do you worry about in situations where you treat with antibiotics, you stop, an infection comes back, and then you treat with antibiotics again? What would you worry about then? What? I can't hear it. No, not necessarily. Like what do you worry about repetitively treating bacteria with antibiotics? Resistance. Like I was starting to worry about like I was like looking at my arm and I was like, this thing is not going away. My antibiotic regimen is almost out. Like, should I ask the doctor for more, or should I just be like, no, I'm kind of being paranoid? And then I was like, but what's going to happen if I don't kill them all and there's still some left, and then I come back and I have to treat? They're going to be resistant then at that point. So I was like going through like all these thought processes, and and the craziest thing was when I went off the 2-week cycle, I immediately got sick again. Like, it went back to feeling how it originally felt. And then after like 3 days, it started getting a little bit better. And so it's like, so my conclusion was like, I really felt like there's no way that these antibiotics killed all the Borrelia in my body. I felt like there was still Borrelia in my body. And eventually I got over it. So like after like 3 months, I finally felt like I was back to normal. But I never went back, and I never asked for like a second cycle of antibiotics. I think I like kind of got over it. But the thing with Lyme disease is Lyme disease has these weird rumors about it. They're not necessarily unsubstantiated, but they're uncorroborated, like they're not proven. And some of the things that people think happens with Lyme disease is they think it never really goes away. So they think that it hides out in your joints, and maybe when your immune system is weakened again, it pops up again later. And there's people who late in life have like neurological issues, like they're psychotic, or maybe they're perhaps having schizophrenia or something like that. They're having all these brain issues, and many people think that it was like this Lyme disease that they got when they were like a kid. And so like it's like a common scapegoat of old people going, like, I don't know about going insane, but it's like a scapegoat that people think might cause their neurological issues. And so doctors always, in many places, are like, well, I don't really know if that's true, or I don't know if this person is just a crazy person, or I don't know if it actually is Lyme disease. But my experience of it made it seem like there probably was still some Borrelia hanging around. And I haven't had any issues since then, but just the strange fact that I got sick again after the antibiotics was what made me think that. Okay, so that was my story with Lyme disease. So doxycycline is the treatment for adults with Lyme disease. If you're a kid, they'll give you something different. Doxycycline is similar to tetracycline. It's a protein translation inhibitor. Amoxicillin was what they give to my son, probably in a similar class. So again, sometimes the risk of treatment is so low, like if you think you have it, you might as well just treat yourself for it. Was kind of my thought. Okay, so let's talk about the life cycle. So let's see, where's the tick eggs? Here's the tick eggs. Let's start with the tick eggs. Here's the tick eggs. They hatch into the larvae. Larvae have 6 legs because we're in acari. So acari are strange in that the larvae of mites and ticks have only 6 legs. The larvae attach to mice and feed on their first blood meal. And in this case, if you're in an endemic region, the mice are the amplifying reservoir. So what is the amplifying reservoir? The amplifying reservoir is the source of the pathogen that gives it to the vector and amplifies the population of vectors being infected by this pathogen. What? No, I'm saying mice are the amplifying reservoir. But I'm saying an amplifying reservoir is a reservoir that has the disease and it pumps up the population of vectors that are infected with the disease. So the ticks are getting it from the mice, not the deer. The deer don't carry the Borrelia. It's the mice that carry most of the Borrelia. Okay. So then they feed on the mice, and then they fall off and they molt. Then as larvae, they're looking for mice, but they will feed on larger vertebrates. So they'll either find another mouse, or they'll find a human or a dog if you're unlucky. And that was the case for me. Was my dog was bringing them in, and during the day, she was going to lay on my bed, letting the ticks off in my bed, and then I would go to sleep, and then they would feed on me at night, which is pretty disgusting. Okay, so that's usually how humans get it. If the larvae feeds on a mouse, it will molt to then adult, or if it gets a blood meal, it will molt to an adult. And then these adults look for and feed on the deer. Okay, so when you say deer tick, it's usually only the adults that are feeding on the deer. Although the larvae will feed on deer if they can get on one. Okay. We talked about the 2 endemic foci, and now you kind of see Lyme disease all over. And here is a picture showing Ixodes ticks. We have Ixodes ticks and we have deer in Alabama. We do have Lyme disease in Alabama, but it's not as high. Why is it not as high? The mice that they're feeding on is different. The mice we feed on is different, and the reservoir in the north is more of an amplifying reservoir. It's more of a competent reservoir for Lyme disease. So we do have it here, but it's at much lower efficiency. Why don't they have in the Pacific side on the west, they have a tick called Ixodes pacificus. Why don't they have much Lyme disease on this side? Similar thing. It's going to be different reservoirs. And I've also heard that ticks on the East Coast feed on lizard, or on the West Coast, ticks on the West Coast feed on lizards. And I was told that some of the lizards have borreliocidal blood. So even if they got it, like some of the reservoirs are just not as competent on the West and in the South as they are in this region. Okay. So in terms of the epidemiology of the disease, deer play an important role, okay, but they're not the reservoirs. But they play an important role because if you have high populations of deer, you also have high populations of ticks. And if you have high populations of ticks, you're going to be encountering them day after day after day. So an interesting thing is that in the 18th and the 19th centuries, we went through a phase where as we were expanding and building suburbs and things like that, we started deforesting the United States. And there was a big drop in the deer population. So they were being hunted and they were also seeing their habitat lost. And during these periods, purportedly, Lyme disease was very low, although they wouldn't have known it was Lyme disease because it wasn't described until like 1976, but they would have, but in terms of like people looking back and finding for things that might possibly be Lyme disease, purportedly incidence was very low. So it was always thought that if you kind of like control your deer populations, you can do a lot to sort of help your community have less Lyme disease. Removal of deer can eradicate Lyme disease. And there was actually a study, it's actually a really cool paper that looked at this, Journal of Medical Entomology. This is the first author. They looked at this hypothesis on this island off the coast of Maine, which is called Monhegan Island. And this was a 13-year study. So every year they would go to this island and they would do a couple things. They would drag for ticks. Yeah. They would measure how many ticks are on rats they would catch, and they would measure how many ticks are on deer they would catch, and then they would do PCRs to check for Borrelia in the ticks over 13 years. And while they were looking at this, something weird happened, and the deer population went extinct on the island. So this is a very small island. And you can see here the deer, there were like 105 deer, and each year the deer got less and less and less and they went extinct. So this study like randomly happened to capture looking at ticks, Borrelia, and deer in a population on an island when the deer miraculously went extinct. And what happened when the deer went extinct was the ticks, so these are adult ticks, flagged from, is it from deer? flagged, that means they were dragged. So these are amount of ticks that they were able to drag year after year after year. And after the deer population went extinct, what happened to the tick population? They also went extinct, although it's not zero. So maybe there's still a couple there, probably feeding on rats and stuff. But this is essentially an important figure that shows you if you get rid of deer, the ticks die too. So it's a good way to control for some of these tick populations. If you can control sort of like hunting through, what do they call those? Department of, well, game and wildlife. Yeah, game and wildlife. They can institute rules that allow for population control of deer, and that can contribute a lot to sort of like Lyme disease. You keep your deer populations in check. Just a quick on how you sample for ticks. There's sort of, you can either drag or you can trap. So tick dragging is when you literally just take a white blanket or a flag and you literally just drag it through the woods. And usually they'll go to like the edge of the woods and drag. And then you just take the white blanket and you can count how many ticks are on it. Yeah, that's the trapping method. So you can also take, Michaela used to do this. So what she suggested was you take like a white sticky trap, and then they would put like a cooler with dry ice and some holes, and then the cooler would like emit the dry ice, and then I assume the ticks just crawl onto this, and you can just kind of like find them. So you can trap and you can drag for ticks. So more about the ancestry of this disease. If you look at Borrelia, and I always spell this wrong, should be that. If you look at Borrelia, Borrelia is not just in the United States. It's not just in New England. You also find this disease you find Borrelia in Sweden, and you find it in Europe, okay? But there's something weird about the Borrelia population in Europe compared to the Borrelia population in the United States, where the Borrelia population in Europe doesn't tend to give arthritis. So there's some genetic difference that has happened in the split between Europe and America that has caused the population in America to cause this arthritis. And we think that from the most recent studies that I've looked at, it's thought that the ancestry of the Borrelia comes from the ancestry of our strains in Minnesota and in New England. It's thought that they come from Sweden. I get that right? Where's this Sweden? This is Norway. This is Sweden, and this is Finland, or this is Finland. I don't know. I think this one is Sweden. It's thought that the Borrelia comes from Sweden. So one hypothesis, maybe, I'm just throwing this out there. Who knows? Maybe, okay, I should also say, it's also, the divergence is not so great that they separated like when Pangea separated. Like they've somehow crossed recently in like human history, okay? And so it's thought maybe, who knows, maybe the Vikings brought it over when Swedish Vikings came. And I don't know, it's interesting because many of the populations in Minnesota and especially like the Midwest are settled by like Swedes and Norwegians. Anyway, it's not that it comes from Sweden. How would you know this? So how would you design an experiment to test for this? That's an open-ended question. How would you be able to conclude like, oh, our Borrelia populations probably come from Borrelia from Sweden? How would you figure that out? What would lead you to that strange conclusion? Yeah, you'd sequence their genomes. So you'd sequence populations of Borrelia. I spelled it wrong. There's only one L. Borrelia in Europe, from different regions of Europe. Let's say you have a Sweden population, another population, A, B, C, and this will be my last point. I'll finish up here. And then you have populations in Minnesota, populations in New England. Okay. And if after you've sequenced their genomes, the populations in Minnesota and New England are closest to this one, but they're very distinct from, they do not equal B, and they do not equal A, and they do not equal C, but they're most closely related to the one from Sweden. That's what makes you think, okay, came from Sweden. That's how we know that. So we sequence their genomes, and you can do what's called MLST, multi-locus sequence tagging, which is looking at SNPs, looking at genetic differences. And it's hoped that if we can discover if we can understand all the genetic differences between different populations, then maybe we can also figure out what it is in our populations that make our Borrelia cause arthritis, as opposed to not causing it in European countries. Okay, so I'll end the Lyme disease lecture there. And the next lecture is going to be on antigenic variation, which is an interesting technique that bacteria, including Borrelia and other pathogens like Plasmodium and trypanosomes, it's a technique that they use to sort of avoid and hide in the immune system. So that will be next time.